Hi everyone, I thought I'd take just a short bit of time here to talk about some of the ratios that you commonly see when we're talking about genetics. And these are ratios, of course, you'll probably come across on your upcoming midterm. So when you are presented with a problem, break the question down, look at the different traits that you're seeing, the different phenotypes, colors or shapes or whatever, and try to figure out a ratio. If you see a 3-to-1 ratio, you're probably dealing with a simple monohybrid cross. If you see a 9-to-3 to 3-to-1 ratio, you're dealing with a dihybrid cross probably. So that involves two genes. If you see a 1-to-2-to-1 ratio, you're dealing usually with codominance or incomplete dominance. 9-to-3-to-4, that's going to tell you there's probably some form of epistasis going on. That's where you have two genes and one gene can override the effects of the other one. And then finally, if you see a 2-to-1 ratio, that tells you that some of the offspring are missing, probably because some of the combinations are lethal. Let's start with the simplest case. That would be a monohybrid cross. Mono means one, so we're dealing with one gene, and that gene has two alleles. There could be more, but for our questions, I'm not going to do more than two alleles. The classic example is with Mendel's peas. If you remember from the lecture, Mendel looked at peas that came in two flower colors. Some were purple and some were white. And he was able to show that the allele for the purple color overrode the allele for the white color. Now, if we take true breeding parents and breed them together, we get all purple plants. What does true breeding mean? Well, that means if we keep taking those individuals and mating them amongst themselves over and over and over again, we always get offspring that look just like the parent. So we can have purple plants that are true breeding, and we'll always get plants with purple flowers. We can have white plants that are true breeding, and we'll always get offspring that have white flowers. Now, if we take these two different true breeding parents and we bring them together, again, we get an F1 generation, a filial 1 generation that is all purple. If we take those plants from the F1 and we interbreed them, then we get a mix of purple and white flowers. We get a 3-to-1 ratio, 3 times as many purple plants as white plants. And the reason for that is that the purple flowers have an allele that is overriding the white allele. We only see the white color if we have two recessive alleles. So the purple is dominant. Remember that we can use a Punnett square to figure out outcomes, or to try to work backwards and figure out the genotypes of a plant we're unsure of. The plants that we see here, the pea plants, are diploid, and they produce haploid gametes, sperm and egg. And what you're doing in a Punnett square is you're crossing those haploid gametes together to see what the diploid zygotes are going to be. So we have our white flowers that are homozygous recessive, and they can only provide the recessive allele in their sperm or egg. We have the purple flowers. The true breeding ones are homozygous dominant. They can only provide the dominant allele. So when we cross those together in our first generation, we get all heterozygous individuals. They have one dominant and one recessive allele. When we mate these amongst themselves, then what happens is we get a mixture because, of course, these heterozygous individuals can donate either. There's a 50% chance that their sperm or egg will contain the dominant allele and a 50% chance that they'll contain the recessive allele. And we look at that second cross there in the F2, and we can see that we get this 3-to-1 ratio. Now, one of the purple plants will be homozygous dominant, but there will be two that are heterozygous. And then, of course, we'll have one homozygous recessive. That's all well and good for true breeding strains that we've bred for many generations and they always maintain their color. But what if we have just some plants that have kind of grown wild in the garden and we're not sure what their genotype is? So here we have a mixture of plants, some that are producing purple flowers and some that are producing white flowers. We do know the genotype of the white flowers. There's only one thing they can be. They can be only homozygous recessive. They have to have two recessive alleles. But the purple plants could be either homozygous dominant or heterozygous. We just don't know because remember, if they're heterozygous, the dominant allele will overpower the recessive allele. But we can do a simple test to figure out what the genotype of those unknown purple plants is. We can take them and we can cross them with a white flowered plant that we know is homozygous recessive. This is really important if you are breeding plants and you come across one in the wild and you want to develop a pure strain of it, or let's say you're breeding animals, rabbits, whatever, and you want to know what their genotype is. This is a very useful process and it's known as a test cross. So if we have an unknown individual and that unknown individual turns out to be homozygous dominant, and we cross that with a homozygous recessive individual, we will get all heterozygotes and they will show the dominant trait, purple in this case. If our unknown individual is heterozygous and we cross that with a homozygous recessive flower, we're going to get half purple and half white. So half show the dominant trait and half show the recessive. So this is a 1-to-1 ratio. And again, this is called a test cross because we're testing to see what the genotype of our unknown is. And we can do this with any dominant trait. So here you're seeing it with a round seed, that's a dominant trait, and we're trying to figure out if our round seeded plant is heterozygous or homozygous dominant. Again, if it is a heterozygous individual, we get a 1-to-1 ratio. Now, if you're a breeder, you do like to keep pure breeding strains, so that's probably not what you want. You'd rather have a cross that will give you all heterozygotes. Now let's move on to a dihybrid cross. Di means two, so we're dealing with two separate genes here. The genes are going to have only two alleles for all of our examples. We have 4 possible genotypes, again because we have 2 genes and they each have 2 different forms, 2 different alleles. So here we've got an example where again we have 2 true breeding strains. We're looking at the shape of the pea. Is it round or is it wrinkled? And we're looking at the color. Is it yellow or is it green? And for this particular set of traits, if we are homozygous dominant for both of the genes, the R gene and the Y gene, then we're going to get a yellow, smooth or rounded pea. If we have an individual that's homozygous recessive for both of those genes, we're going to get peas that are green and wrinkled. If we cross them together, we're going to get a generation that is heterozygous for each of those traits. So big R, big Y, little y. It looks like the homozygous dominant parent. But now if we take that generation and we breed it amongst itself, then we get an interesting mix of traits. Again, we get 4 different outcomes. So what we're going to do when we do this, we're starting with a diploid individual. And remember, the gametes that we draw on the axes of the Punnett square aren't diploid. They have to be haploid. They can only have 1 copy of an allele for each of those traits. So we could have a big R and a big Y from that heterozygous. That's what it could contribute as a sperm or an egg. We could have a big R and a little y. We could have a little r and a big Y. And we could have a little r and a little y. That's the 4 possible outcomes. We do the exact same thing on the other side. So we're looking at the male gametes and the female gametes. And when we cross them, we just write in the spaces, the cells of our Punnett square, those combinations. So if we have big R, big Y crossed with big R, big Y, of course, we're going to get big R, big R, big Y, big Y. So always remember that what you put on the axes, that has to be haploid. There's only going to be 2 letters. And the outcome that you have in the Punnett square is always going to be diploid. There's going to be 4 letters. So you can see that there's 9 ways that we can get a situation where we have individuals that look like the homozygous dominant parent. They're not all homozygous dominant, of course, but they look like they are. They have the dominant traits. There's 3 ways that we can get individuals that are green, that's a recessive trait, but also round, that is a dominant trait. And there's 3 ways that we can get the opposite. Individuals where we have the wrinkled state, which is recessive, but we have the dominant color. And then there's one way, only one way out of the 16 possibilities, where all the alleles can come together such that we have recessive alleles for everything. And of course, that's going to give us a green wrinkled individual. So look for this 9:3 to 3:1 ratio in the second filial generation, the F2. If you see that, and we started with true breeding strains, then we're likely dealing with a dihybrid cross. Alrighty, let's have a look at codominance and incomplete dominance. We're dealing with 2 true breeding strains once again, and we're only looking at a single gene with 2 alleles. However, unlike we saw in a simple monohybrid cross, we don't have only 2 phenotypes, we have 3. This here is an example of incomplete dominance. Codominance looks a bit different. I'll talk about that in a second, but it has the exact same ratio. We're starting with 2 true breeding strains. One is white, one is red. The white one is homozygous recessive. The red one is homozygous dominant. When we cross those parents, we get a new phenotype in the F1 generation. All of the flowers are going to be heterozygous, of course, but they're going to be pink. If we take those pink flowers and we mate them amongst themselves, in the F2 generation, instead of getting a 3:1 ratio, we get a 1:2:1 ratio. We have one of our 4 plants, white, so it's homozygous recessive. There's going to be one that's red, homozygous dominant, and there's going to be 2 heterozygous individuals that are pink. So the heterozygous individuals in this case are kind of a blending of the 2 original parents. And note that we could do a test cross on our F1 generation, on our heterozygous individuals. And when we do that, we're not going to get any red adults. Instead, we're going to get 1/2 pink, those are the heterozygous individuals, and we're going to get 1/2 white, those are the homozygous recessive individuals. So how does incomplete dominance differ from codominance? Again, we have the same ratios. It's just the phenotypes are expressed a little bit differently. Starting on the right there, we have an example of incomplete dominance, and it's exactly the same as we saw in the snapdragons just a moment ago. The heterozygous individuals are a blend of the pure breeding phenotypes. So we have pure breeding red, pure breeding white. Bring them together, we get pink. On the left, you can see an example of codominance. In codominance, again, the same ratios, but in the heterozygous individuals, both of the pure breeding traits are seen. So here we have a cow that's kind of beige in color, and we're crossing it with a brown cow. And instead of getting a light brown cow, we get a beige cow with brown spots. So we can still see both of those phenotypes. They're not blended together. Instead, it's kind of patchy. Both of those phenotypes are being expressed in the heterozygote individual. Here's another nice example of codominance, although this one is a bit trickier. We're only dealing with one gene here, but this gene has 3 possible alleles. I did mention before that I wouldn't do that to you. I guess I lied. This is one case that I do want you to be familiar with where we have more than 2 alleles for a gene. We're looking at blood typing here, specifically ABO blood typing. On your red blood cells, you have lots of proteins poking out. And we're looking at one particular set of glycoproteins that is coded for by one gene. Remember, a glycoprotein consists of proteins with sugars attached. We have the A form of the allele that has certain sugars attached. We have the B form of this gene that has certain sugars attached, and they differ from what we see in the A allele. And then we have type O, where we're actually missing those sugars altogether. If, let's say, your mother donated the allele that's type A, and your father donated the O allele, your blood type would be type A. The A is dominant over the O. If your mother donated the B allele, and your father again donated the O allele, you would be type B, because B is dominant over O. So A and B are both dominant alleles, and O is a recessive allele. The only way that you can be type O is if you get an O from both parents. You have to be homozygous recessive to get the O. If you got an A from one parent and a B from another, you would be AB. So you're heterozygous, but you have both types of glycoprotein, and they both have an impact. So if you were to donate blood to someone, and let's say that person was type O, their body is unfamiliar with both of those proteins, and they would attack both of those proteins. So I won't get into that in great detail right now. I think I did talk about this a bit more in our longer lecture, but this is important stuff. Of course, you need to know what blood type you are to determine who you can give blood to and who you can receive blood from. Essentially, you produce antibodies that attack the glycoproteins you don't have. And if you receive blood with those glycoproteins, that's a problem. It causes your blood to agglutinate. It causes your red blood cells to stick to each other. And as you can imagine, that's not going to help them get through your circulatory system. That's a bad thing. Now, on the left, I've showed you my preferred way to write these different genotypes. On to something different. Let's take a look at epistasis. We're looking at an example here where we've got 2 genes, but one gene overrides the effects of the other gene. Think back to the dihybrid cross. In the examples we were looking at, the genes were independent. One gene did not affect the outcome of the other gene. But here, we're usually dealing with a biochemical pathway. That's what you're seeing with these mice here. We're looking at a biochemical pathway that has 2 steps to it. There's two different enzymes, one that's used for each step. One enzyme, the very first enzyme in the pathway, is coded for by the C gene. The second enzyme in the pathway is coded for by the B gene. We start out with a compound that's colorless, and the C gene produces the enzyme that takes it and changes that to make it a pigment with color. So the first gene, again, is the C gene. If we have at least one functional copy of that gene, we can get through the first step. The second step is controlled by the B gene, and there's two alleles of the B gene. If we have the dominant allele, so the big B, then we get very dark pigment. We get black coloration. If we have the recessive form, it's not totally broken, but it doesn't work as well. We get some pigment building up, but not nearly as much, and we get the brown coloration. So once again, the C gene starts the process, and then the B gene finishes it. Now, if the C gene doesn't work at all, it doesn't matter if you've got a fully functional B gene. The color is still going to be white. It's going to be colorless. There's not going to be any pigment. So we have to get through that first step to get any pigment whatsoever. And then, of course, the condition for the second gene, whether or not you have the uppercase B, is going to determine whether it's black or whether it's brown. If we do crosses in this case, we don't get that 9:3 to 3:1 ratio. We get a 9:3 to 4 ratio. So think back to the dihybrid cross. That 1 in the 9:3 to 3:1 was an individual that was homozygous recessive for both genes. But here, any situation where we have two recessive C genes won't give us any pigment whatsoever. So we get 4 instead of 1 individual that shows those two recessive traits in the F2 generation. Let's have a look at another example. This is coat color in Labrador retrievers. So Labrador retrievers come in 3 colors. We have black and brown, and that's due to different forms of a pigment known as melanin. And then we also have yellow retrievers, and they don't have this melanin. I have a brown retriever. She looks just like the one with the red dot on her chest. I have no idea why that red dot is there, by the way. But anyway, what's happening here is once again we have one gene that overpowers or overrides the effects of another. What's going on here is a little bit different from the last example. We have an E gene that activates melanocytes. Melanocytes are cells in the skin that produce melanin, that brown or black pigment, and that pigment accumulates in the fur to give the fur its color. If the dog has at least one active form of the E gene, so they have at least one dominant allele, then their melanocytes will be active and they'll be capable of making melanin. The gene that determines the color of the melanin comes in 2 alleles. We have the dominant allele, which will give us the full black color, and the dog just has to have, at least one of those alleles. And then we have the little b, the recessive form. If the dog is homozygous recessive for the B gene, like my dog is, then it will be brown. So again, the E gene is going to override the effects of the B gene. If the E gene is not working, it doesn't matter if you have 2 fully functional forms of the B gene, or only 1, or you have 2 recessive forms. the dog is going to end up yellow because that's the color of the fur when there's no melanin whatsoever. And again, we get this 9:3:4 ratio because that C gene is so important and it overrides the effects of the B gene. Finally, let's look at the sad case of homozygous lethal traits. It's exactly as the name suggests. With this kind of inheritance, the homozygous recessive or the homozygous dominant, depending on what gene we're talking about, is lethal. If we're talking about mammals, they don't survive till birth. They might abort very, very early on in development. So here we have a case where we're taking two heterozygous mice, and they have a yellow coat, as you can see, and they're fine, they're healthy. If we mate them, we would expect, of course, to see 2 out of the 4 offspring heterozygous, and they're yellow, they look like the parent. We would expect to see 1 of the 4 being homozygous recessive, and in this case they're also fine, but they're white. And we would expect to see 1 out of 4 with the homozygous dominant trait. But unfortunately, those mice don't survive. They abort very early. So if you were to breed these mice at home, you wouldn't see that genotype. So you would get a 2:1 phenotypic ratio. In class, we also talked about pedigrees. And pedigrees show how traits are passed down from generation to generation. Here's a pedigree here, and we've got the generations numbered. Generation 1 gives rise to generation 2, gives rise to generation 3. And we can use pedigrees to look for inheritance patterns within a family, for instance. So just a few reminders. Males are shown as squares. Females are shown as circles. If the square or circle is filled in, it means they have the trait that you're interested in. Usually it's a bad trait, hemophilia or something like that. We looked at 5 different inheritance patterns: mitochondrial, autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. And I've included next to those words the types of genotypes you might have. Now for mitochondrial, we're talking about the genotype of the mitochondria. Remember, in humans, this isn't the case in all mammals, but in humans, we inherit all of our mitochondria from our mother. So the zygote gets all of its cytoplasm from the mother. The mitochondria from the father's sperm do not contribute. They stay attached to the sperm. Now this is kind of interesting. Mitochondria, if you remember, came from bacteria. Bacteria are haploid. So we use either uppercase A or lowercase a generally for mitochondrial traits. With mitochondrial traits, if the mother has the trait, so the mother has a defect in one of the mitochondrial genes, that trait is passed down to all of the offspring, male and female. Autosomal dominant, you can use uppercase A and lowercase a, or pick your own letter, whatever you want, but that's the format we would use. And what we're talking about here are traits that pop up in the dominant form. So the heterozygote or the homozygous dominant. Autosomal recessive traits are only going to show up in the homozygous recessive individuals. Now on to X-linked dominant. So here we're looking for differences between sexes. Does a certain trait pop up more in males than females, for instance? And remember, when we're talking about genes that are on the X chromosome, we use an X to represent the X chromosome, and then we would put a superscript, big A or little a. If we're talking about a male, they are XY. Don't put the trait, the big A or little a, on the Y. We're only talking about genes that are on the X. The Y does not contain a whole lot of information. It has a few genes that determine whether or not you're going to be male, and of course also help influence sperm production, but that's it. Now, if we're talking about X-linked recessive, that is going to be a trait that is far more likely to show up in males. And the reason for that is males only have one X chromosome. If you're female and you get one X chromosome with a bad gene, you might be okay because maybe your other X chromosome doesn't have that broken gene. It has the dominant form of the gene. But if you're male and you have this trait in your family, you're more likely to express that gene, to show that trait. Here are the steps I would suggest you take to try to decipher a pedigree. I think pedigrees are kind of fun. They're like little mysteries that you have to solve. So the first thing to look at is, are males and females affected equally? If yes, you're probably dealing with a mitochondrial or autosomal trait. Remember, autosomal refers to all of the chromosomes except for the X and Y. If not, if there's a disparity between the two, especially if there's more males affected, then you may be dealing with X-linked. And if it's X-linked, then we're going to skip to step 4. We'll come back to that. But let's assume that it's autosomal. First thing to check for, the easiest thing to check for, is it mitochondrial? If it's mitochondrial, then all of the offspring, male and female, of an affected mother will also be affected. And to check to make sure this is the proper inference, check to make sure that the trait is never inherited from a male parent. Males can't pass down the mitochondria in humans, so you should never see that if it's mitochondrial. If this doesn't fit, we proceed to step 3. We assume that it's autosomal. Now we have to determine if it's autosomal dominant or autosomal recessive. If it pops up every generation and it's quite common, it's likely to be autosomal dominant. Now, how do we check this to be absolutely certain? Every affected child must have at least one affected parent. And also, both parents of the affected child might exhibit this trait. If autosomal dominant doesn't fit, let's try out autosomal recessive. And there's one really good way to check for this. If the trait is observed in offspring, children, that lack an affected parent, then yes, we know it's autosomal dominant. Again, if none of the other traits fit. Why is this? Well, imagine that the parents are both heterozygous. They would not show this trait. They could each pass down a recessive allele, though, and we could have children that have 2 recessive alleles, one that they got from each parent. They would be autosomal recessive. They would show the trait even though their parents did not. All right, now what if this trait pops up more commonly in one sex than the other? Then we're probably dealing with an X-linked trait. And now we have to decide if it's recessive or dominant. If it's recessive, the check for this is that mothers will pass the trait to all of their sons. So the mothers will pass X chromosomes to their children, and only X chromosomes. Fathers will pass one X or one Y. It's a 50-50 chance. So if a mother is X-linked recessive, she shows a trait if she is homozygous recessive. The sons got their Y from the father, so they had to get their X from the mother, and in this case they would have to get a recessive allele, and they don't have a dominant allele to override that. Finally, X-linked dominant. If it's X-linked dominant, one key thing to look for is that fathers will pass it to all of their daughters. So if a father has, let's say, X, big A, Y, the daughters are going to get one X from the father, one X from the mother. They're going to get that dominant X from their father. They are going to show this dominant trait. Affected mothers, they can pass that trait to any offspring, so daughters or sons. Okay, so I hope this quick little review of the genetics was helpful. Please let me know if you have any questions.